Thanks so much. This has been a really fun event for me. I'm learning so much, and it's really nice to be exposed to kind of the range of both research and more technical side of things. So today I'm going to tell you about research in my laboratory on the role of inhibitory neurons in auditory processing. And to take you kind of out of the visual world and into the auditory world, if the sound works, I'll play you a little clip of a piece by Rachmaninoff. So as you were hearing this beautiful piano piece, your brain was performing multiple computations, the most basic of which are frequency discrimination, so telling apart the difference in the frequency of the sounds that you hear, and another one is putting together the string of notes into a melody. Those are just some of the computations that your brain makes when listening for sounds, and they're important not just for music perception, but also for speech perception, communication more generally, auditory navigation. The goal of research in my lab is to try to understand the neuronal mechanisms in the brain that make sound processing, and especially complex sound processing, possible. And we want to understand that at the level of neuronal circuits. In our lab, we have been focusing on trying to understand the function of the brain region called the auditory cortex. This is a little different from the visual system. In the auditory system, the auditory cortex is actually the sixth synapse from the cochlear, so it's a little higher up in the auditory hierarchy. And it's thought to be the key region where the representation of sound is transformed from representing the physical sound features of the waveform, such as, for example, the frequency of the notes, into a more object-like representation. So your kind of perception of the musical piece. And what I'm going to tell you today is that within the auditory cortex, distinct types of inhibitory neurons, which luckily for me, Andreas already introduced so beautifully, play different roles in many, many aspects of sound processing. And so in our research, we have been focusing on trying to understand the functions of these neurons from multiple points of view, both trying to understand their function in auditory perception and computation, and also in shaping network dynamics. I've been interested in understanding inhibitory excitatory interactions ever since my PhD, actually just down the road at Harvard, where in the salamander retina, I studied the function of inhibitory neurons in visual perception, very much like what Andreas told you about these more complex shaping more complex receptive fields and contextual processing. From a computational perspective, we have known for a long time that a network that's comprised of excitatory and inhibitory units, even with very simple connectivity processes, can give rise to a host of very interesting and complex behaviors. And over the last decade, we have come to appreciate that if that network is comprised of not just basic excitatory inhibitory neurons, but different types of inhibitory neurons, that added complexity can actually produce an added complexity in the types of computations such a network can perform. This branch of research in my lab, where we were looking at the interactions between excitatory and inhibitory populations, we were kind of interested in bringing together what we know about the computational possibilities in these networks and trying to design experiments to put some computational constraints on building a realistic cortical models of these interactions. So I'm going to go a little fast since I want to catch up on our time, but I'm going to tell you about 4 different findings in the lab. First, I'm going to tell you that cortical inhibition actually controls frequency discrimination. Different inhibitory neuronal subtypes differentially control adaptation to temporal regularities in sound. I'll tell you about a circuit model that we built based on results from ours and other laboratories. And I'll also show you that a recent study where we find that different inhibitory neurons actually shift neuronal dynamics between different modes of representation. So when we initially started this research program in the lab, we wanted to go back to the most basic sound behavior, which is frequency discrimination. And you can think of it, I'm sure some of you in this audience have perfect pitch and can tell very small differences in sound frequencies. Or maybe some of you are more like me and completely tone deaf. For example, I can't even tell whether one note is higher or lower frequency than the next. This is something called frequency discrimination acuity. It varies across individuals. And what we were able to show is that, in fact, it's controlled by inhibitory neurons in the auditory cortex. So we built a very simple behavioral test for a mouse where we could measure frequency discrimination, how well it could detect difference between two notes. And we postulated that if inhibitory neurons control this difference should have something to do with the degree of frequency tuning of neurons in the cortex, which are comprised through some combination of excitatory and inhibitory components. By modulating the activity of inhibitory neurons, by either up or down regulating them using either a channel inactivating or a suppressive opsin, such as channel rhodopsin and archirhodopsin, we predicted that we should be able to shift this frequency discrimination acuity in the opposite directions. So upregulating parvalbumin inhibitory neurons should increase inhibitory drive, narrowing the frequency tuning, and thereby shifting the frequency discrimination acuity up, so improving acuity. And we expected the opposite effect with archirhodopsin. And I wouldn't be probably telling you about this study from, almost 10 years ago now if it weren't in fact true. So when we modulated differentially the activity of inhibitory neurons, we were able to either improve or impair frequency discrimination acuity. And these changes could be predicted based on the changes in neuronal activity in the auditory cortex. And as a control, we showed that simply upregulating the activity of excitatory neurons, which is one of the consequences of shutting down inhibition in the cortex, did not in fact change frequency discrimination acuity or tone response magnitude in the brain. So, all right, so this was very exciting because now we learned that inhibitory neurons actually control a very basic auditory computation. But there are many different types of inhibitory neurons, and next we wanted to understand more specifically whether these different types of inhibitory neurons play a differential role. And in particular, we were really interested in understanding the difference between a class of neurons that I already told you about, parvalbumin neurons, that target the cell bodies of excitatory neurons, and another class of neurons called the somatostatin-positive interneurons that target the distal dendrites of inhibitory neurons. And in fact, a number of laboratories has tried to see whether they play differential function and frequency discrimination, and there wasn't really a strong differential effect. And so we wanted to see whether these differences in there could actually support auditory adaptation to statistical regularities. So this is this is a feature of neurons in the auditory cortex. Almost every neuron that you record from, 95% of the neurons, respond to sounds differentially based on the statistical regularities with which those sounds are presented. And it's usually studied with a very simple stimulus where you have tones of two frequencies, one of which occurs frequently and the other one is presented rarely. A priori, these two tones we record from neurons which are equally responsive to these two tones. But when one of these tones is overrepresented, the response to that tone goes way down, and when it's overrepresented, the response to that tone increases. And when we switch this relationship switches. So the cortical neurons respond strongly to unexpected sounds, and their response adapts over time to sounds that are repeated. This is a really important aspect of cortical computation and more generally auditory computation, because it allows the brain to extract statistical regularities in sounds and also make predictions for expected or unexpected sounds that would be coming. And it's traditionally thought as a form of notched adaptation. Some of the aspects of this adaptation have been accounted for by a feed-forward model, where more narrowly tuned inputs adapt over time, and so the adaptation at the cortical level comes from the already adapted inputs. We wanted to test the hypothesis that there is an additional component to this adaptation through the inhibitory neurons. And long story short, when we inactivated the parvalbumin and the somatostatin neurons while testing for stimulus-specific adaptation, we found that without the inhibitory neurons, that adaptation was without inhibitory neuronal firing, that adaptation was really strongly reduced. But this effect was differential, where if we tracked how that reduction happened over the time course of adaptation, the contribution of somatostatin neurons increased as the network activity adapted to repeated tones, whereas the suppression of parvalbumin neurons produced a more uniform effect. So, in fact, the study found that the somatostatin positive inhibitory neurons were actually specifically supporting adaptation to temporal regularity in sounds. At the same time, many laboratories were trying to understand the function of these neurons in different forms of auditory processing using a number of different paradigms. So we, with the help of a talented postdoc, Youngmin Park, we decided to put together the results of these studies and build a functional model for inhibitory excitatory interactions in the auditory cortex, trying to constrain the parameters in that model such that it would reproduce the results that were experimentally observed in our study, and then study from several other laboratories in different paradigms. As you can see, even if we just include two inhibitory neuron types in this model and then excitatory neurons, trying to account for all the different synapses between them is actually somewhat a daunting task. And so we did a parameter sweep over these biologically controlled parameter sweep over the parameters for the connectivity between the inhibitory and excitatory neurons. And what we found was the key two aspects of synaptic connectivity that accounted for this range of experimental results was a depressing synapse between the parvalbumin neurons and the excitatory neurons, and a facilitating synapse from excitatory neurons to somatostatin neurons. Now, these are synapses that have these properties of these synapses have been previously documented in the literature, so they're consistent with the anatomy. And so using these models, we were able to actually reproduce the results from our study We were able to make predictions for what would happen if we could instead activated the inhibitory neurons instead of suppressing them. We were able to account for the results in other studies. Here I'm just showing you a study from Andrea Hasenstab's lab that found differential role for somatostatin and parvalbumin neurons in forward masking. And so this model was made available and could be used to make predictions for other types of experiments that people would want to do. More recently, we started thinking about how different types of inhibitory neurons can shape the dynamics, the neuronal population dynamics. Now, something that the auditory cortex and the auditory cortical literature is well known for is that the representation of sounds in the auditory cortex, unlike in the primary visual cortex, is sparse. And so that means that any given sounds drives the activity of a few types of neurons. But this varies somewhat across the species, and in our understanding of how the brain makes use of different types of population coding. There have been several different models for what that could be useful for. And one idea in auditory literature is that sparse codes can lead to localist representations of sounds. That means that different neurons represent different stimuli. In the kind of most extreme case, if you took two sounds, then they would activate non-overlapping populations of neurons. On the other extreme, there is this idea of a distributed code, which is also known as a rate code, where different stimuli would activate similar populations of neurons, but at different rates. And these two forms of population coding can actually be more useful for different tasks that you could imagine. For example, for categorization, you could imagine a localist code could be useful. However, for generalization, you would want a distributed code. And this can also be thought of as can be represented in a multidimensional population space, where we can think of the representations of different stimuli as population response vectors. Under localist codes, two stimuli would be represented by orthogonal vectors, whereas at the kind of extreme in a distributed code, you would have overlapping population vectors at different lengths. So what we wanted to see whether activating different types of inhibitory neurons would actually shift the neuronal representation towards one type of representation or another. And I'm gonna, since we're running out of time, I'll go a little quickly, but essentially what we were able to show is that activating somatostatin neurons actually drives these population vectors away from each other, making the representation more localist, whereas activating the VIP neurons aligns the population response vectors more closely, however, making the changing the vector strength in this population response space. And we did this by measuring the activity of population of neurons using 2-photon calcium imaging and optogenetically manipulating the activity of these two populations of neurons. So let me see. And so this is actually the data that show the changes in the population response angle strength and vector strength. All right, so I hope that I convinced you that cortical inhibitory neurons are really important for auditory perception. And I would like to thank and acknowledge the people who did this work. So Ryan Nathan and John Briguglio were graduate students in the lab who worked on the first set of experiments that show you on stimulus-specific adaptation. Youngmin Park built the computational circuit model, and Melanie Toban and Junki Sheth did the two-photon experiments and analysis.
Any questions while we set up the panel discussion? Go ahead.
Can you cast your results a little bit more to the language of populations or like manifolds? Do you think these neurons maybe control curvature or maybe contribute to some sort of homeostatic mechanism?
Yeah, I actually started writing a grant proposal exactly on that. Does activating inhibitory neurons actually how would that change the relative shape in the population manifolds in encoding different types of sound objects, for example? So in the context of categorization, can inhibitory neurons actually, as the task is learned, shift the population manifolds to be more aligned or more dissimilar? Yeah, that's a perfect question.
All right.
We'll have an answer in 5 years.